Hey everybody, it's time to talk about cell processes. I have broken these notes up into two parts, so they should be two sections of a little less than 10 minutes each for your ease and so your brain does not explode. Speaking of explosions, let's get started. Let's talk about the first topic, which is homeostasis. So we know that all of our cell organelles do different jobs inside of our cells, but the whole point of them doing these jobs is to help maintain homeostasis. Homeostasis means keeping sameness. All organisms want to maintain a particular temperature, level of water, level of sugars in order to survive. For example, even you as a whole person, when you're hot, you sweat. When you're cold, you shiver. That's to try to keep your body at a constant temperature. More on that later. But at the cellular level, all cells are trying to maintain homeostasis as well. They do several things to do so. They need to obtain and use energy. They need to exchange materials. They need to eliminate waste. All of this is to keep the cell happy and at a constant state. One of the most important cell parts to help maintain homeostasis is going to be the cell membrane. Remember, exchanging materials is a vital job the cell needs to do to maintain homeostasis. The cell membrane is the king of this role. So let's go to a quick Amoeba Sisters clip of how this works. There's going to be a couple little added details that I'm going to try to skip through for you to keep it short and sweet. Let's check it out.
Have you ever wondered what it might be like to be inside a cell? Imagine the genetic material, the cytoplasm, the ribosomes. You'll find those in almost all cells, prokaryotes and eukaryotes. Eukaryote cells, in addition, have membrane-bound organelles. All of those structures and organelles have different functions, but cells are not isolated little worlds. They do have a lot going on inside them, but they also need to interact with their environment. It makes sense that to keep a stable environment inside themselves, otherwise known as keeping homeostasis, they must have some control on what goes in and what goes out of them. A very important structure for this that all cells contain is the cell membrane. By controlling what goes in and out, the cell membrane helps regulate homeostasis. It's the homeostasis king. Let's take a look at the cell membrane. You could have a whole course on the cell membrane itself. It has amazing structure. It has signaling abilities. But to stick to the very basics, it's made of a phospholipid bilayer. Bilayer means two layers, so you have these two layers of lipids. Now, these lipids, they're called phospholipids, well, they have a head that is polar, and they have a tail that is nonpolar, making them quite unique. Some molecules, they have no problem going through the cell membrane, and they directly go through the phospholipid bilayer. Very small, nonpolar molecules, they fit in this category. They're a great example. Like some gases, oxygen and carbon dioxide gas, those are great examples. This is known as simple diffusion. Also, it doesn't take any energy to force those molecules in or out, so it's known as passive transport. Simple diffusion moves with the flow, meaning it moves with the concentration gradient. Molecules move from high concentration to low concentration. So when you hear someone saying that something's going with the concentration gradient, that's what they mean. They mean it's going from a high concentration of molecules to a low concentration of molecules. Now, remember how we said the cell membrane is actually a pretty complex structure? Well, one thing we haven't mentioned yet are proteins in the membrane, and some of them are transport proteins. Some transport proteins act as channels. Some of these proteins actually change their shape to get things across. Some of them open and closed based on some kind of stimulus. All of these are good things because it's helping with molecules that may be too big to cross the membrane on their own, or molecules that are polar and therefore need the help of a transport protein. This is known as facilitated diffusion. It's still diffusion, and it still moves with the concentration gradient of high to low. It doesn't require energy though, so it's also a type of passive transport. It's just the proteins are facilitating or helping things pass. Charged ions often require a protein channel in order to pass through. Glucose needs the help of a transport protein to pass through. In osmosis, for water to travel at a fast rate across the membrane, it passes through protein channels called aquaporins. So these are all examples of facilitated diffusion, which is a type of passive transport and moves with the concentration gradient of high to low concentration. Now all the transport we've mentioned has been passive in nature. That means it's going from high concentration to low concentration. But what if you want to go the other way? For example, the cells lining your gut, they need to take in glucose, but what if the concentration of glucose in the cell is higher than the amount of glucose concentration in the environment? We would still need to get the glucose in, so it's going to have to be forced against the regular gradient flow. Movement of molecules from low to high concentration will take energy because that's against the flow. Typically, it's going to require ATP energy. A reminder that ATP, adenosine triphosphate, it has 3 phosphates, and when the bond for the last phosphate is broken, it releases a great amount of energy. Yeah, so ATP is a pretty awesome little molecule. ATP can power active transport to force molecules to go against their concentration gradient. And one way it can do that is actually energizing the transport protein itself. One of our transport. We're still in active transport right now, but let's say a cell needs a very large molecule. Let's say a big polysaccharide.
If you check out our biomolecule video, that's a large carbohydrate.
Well, you may need the cell to fuse with the molecules it's taking in order to bring it inside, and this is called endocytosis. Think endo for in. Often this fusing of substances within the cell membrane will form vesicles that can be taken inside the cell. Endocytosis is the reverse direction of endocytosis. Think exo and exit. They sound very similar too. Exocytosis can be used to get rid of cell waste, but it's also really important for getting valuable materials out that the cell has made. Want a cool example? Well, think back to those polysaccharides. Did you know that large carbohydrates are also really important for making plant cell walls? Cell walls are different from cell membranes. I mean, all cells have membranes, but not all cells have a wall. But plant cells do. And if you're going to make a cell wall, you're going to need to get those carbohydrates that are produced in the plant cell out of the cell to make the wall. So there's a great example of when you'd need exocytosis right there. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Alright, so let's do a quick recap about how that cell membrane actually works. I know that is a lot of information to take in. So let's start with passive transport. Remember, passive transport is when no energy is required to move across the membrane. Remember, passive, no energy. Diffusion is a type of passive transport when small particles like oxygen and carbon dioxide can go with the flow and naturally move from high to low concentrations. This is called diffusion. I wanted you to think about the particles as like students in a hallway. If you have a choice of 2 hallways to go through, one that is jam-packed with students, like those pesky 6th graders who still don't know how to use their lockers, if you had to move through a jam-packed hallway of 6th graders, or if you could easily get to class through an empty hallway, what would you choose? Most likely you're going to choose to go from the crowded hallway of 6th graders to the less crowded hallway so you can get to class on time and not get a tardy. This is called diffusion, wanting to be not in an area that's crowded, but in an area that's less crowded. This is called passive transport because it doesn't take any energy. The particles naturally want to move from high to low. Osmosis is a very special word for when water in particular wants to move across the cell membrane in this passive transport fashion. It wants to go from an area of high crowding of water molecules to an area of low crowding of water molecules. So the special word for water diffusing is called osmosis. Let's talk now about active transport. Active transport is called active because it takes energy. Some of your larger molecules are going to require a lot of energy to move across the membrane. Also, what if you want to move against that concentration gradient? What if you're a rebel and you want to go from a low concentration into a high concentrated area? There are two types of active transports. Endocytosis is when a large particle enters the cell. So endo, taking in. For example, taking in food. When an amoeba wants to take in its food, we learned how it makes little pseudopods that wrap around its food and create a vesicle and pull it in. That's called endocytosis. There's a picture on your paper if you look at your notes. Exocytosis is when large particles need to exit the cell. For example, maybe letting out a large amount of waste. So a vesicle will rise to the cell membrane surface and then need energy to be pushed out of the cell. So these are ways that we actively transport things across the membrane. I know that's a lot to take in, and we've learned a lot of information so far. So why don't you take a little brain break and then head to part 2 when you're ready.
Goodbye. Oh no!
When you're ready for part 2, go ahead and move on. Thank you.